lecture, the recorded version is also up on the Cranial site. So you have that. And it's more of an introduction to how we're going to approach the cross-sections, which will be the major focus in this block. And then the good news is, after this, we start using all of that information to start talking about systems. So we'll start out, like I said, there's a lot of redundancy built into this block. There's a review session scheduled at the end of the block that will do a little quick quiz in class and see how you're doing in that. Some ideas of how to approach. Okay? As I said before, if you took your exam using a paper copy, either here or SODS or whatever, I did bring the paper copies. You can look at those after class. And for those of you who took it virtually, I set up a session today from 3 to 4:30. You can log on. The link is on the Carmen site. You'll go into a waiting room because I only want to bring one person in at a time to look at their specific exams. So we can't spend a lot of time going over the questions, but at least you can see where you missed some points. If you need to have a more serious, longer discussion, then we'll have to set up some separate appointments for those, okay? Which I'm happy to do for you. I may be able to even put some, I can look at my calendar, I set up some in-person, I'll just have some office hours here, but I can look at my calendar again. Let's see how it goes this afternoon. So, we're going to focus again today specifically on nuclei that are related to the cranial nerves. We talked a little bit last week on how you can organize that when you're looking at a cross-section. I mean, you can rote memorize where they are, but I gave you some guidance on how you might approach a cross-section, how you might approach somehow get down to a smaller number of possibilities. And that's what we want to kind of focus on today. So, as Santiago Ramon y Cajal said back in 1909, the brain is a world consisting of a number of unexplored continents and great stretches of unknown territory. And that's where we are right now for you guys. It's pretty unknown, but by the end of this block, you'll know the geography pretty well. I did do a little bit of updating just before I came down, so I put the updated version on the Carmen site. Nothing really major, just a couple of things.
Your projector's off, by the way.
No. Thank you. I don't know why it says it's on. The projector is on. Let me see what we had. Thank you. I still haven't developed eyes in the back of my head to see what's going on. But yeah, anytime something like that happens, just say, hey, not up there. Okay. Like I said, I did make a few minor changes on a couple of slides, but nothing terribly major, but just I reworded some of the objectives in a couple of other places. Okay, so this was what we started talking about at the end of the lecture last time. And this is is there a way to approach, some kind of big picture? How do I identify these structures that we're going to be looking at in the next series of slides? So we talked about in the spinal cord, when we look at it, we know that sensory information coming in through our dorsal root ganglion cells primarily is processed in the dorsal part of the spinal cord, right? And they further divided it in saying that somatic sensory is maybe just a little bit more dorsal than visceral sensory. And then we come a little bit more ventral, and that's where motor information arises. So here's our motor neurons that are going out to supply our skeletal muscles, And again, just a little bit kind of in between is our visceral motor neurons. When we're talking about spinal cord, we would be talking about our preganglionic sympathetic neurons. So that's a nice organization, very simple. Dorsal, sensory, ventral, motor. Unfortunately, when we get to the brainstem, doesn't quite hold up, but there is still an organization. So as we move in, as we move up, especially when we get to the level of the fourth ventricle. So here's my medulla, and let's just say this was my sensory area, and the palms of my hands were my motor area. Now when I get to the fourth ventricle, that opens up. Remember, we call that the open medulla. So what happens is, what was medial, or what was dorsal, kind of gets pushed out laterally, and what was ventral gets pulled up medially. Does that make sense? And then so here's your first really important thing to keep in mind. If we are looking at structures that are located primarily along the dorsal surface of the medulla, pons, or midbrain, the odds are that's going to be related to the cranial nerves. Your cranial nerve nuclei reside pretty much, few little exceptions as always, pretty much along the dorsal surface or the dorsal area of those three regions of the brainstem. So that's going to be a help. If again, if the arrow is pointing at something way down here, don't think cranial nerve nucleus. And then we can further look at this organization by saying, okay, what was ventral is now going to be immediately adjacent to the midline. So that's going to be motor. Again, spinal cord, my palms, I open it up, and my palms wind up on either side of the midline. You can do this on your own. Okay? Here's my spinal cord. Let's push it out. So instead of having now a more dorsal ventral orientation, our various functions are going to take a medial to lateral orientation. Motor will be medial. Think of the M&Ms. Okay? And that means, kind of by default, sensory is going to be more lateral. So if I'm looking at something that's right on the midline, I'm not going to identify it as a cranial nerve nucleus that's involved in carrying sensory information. If I'm looking at something lateral, don't think motor function, don't think hypoglossal, don't think oculomotor. Okay? So get these big concepts, and that's going to help you identify. We're going to work through that today. Most medial would be somatic motor because it had been most ventral. And then we move just a little bit more laterally. We get our visceral motor, visceral sensory, and somatic or special sensory in the case of our cranial nerves, because they have that extra sensory component. Does that make sense? Can you visualize that? Okay, we're gonna put it into play here in a second. Okay, so that's everything I just said. Nuclei concerned with somatic are going to be right along the midline. The exceptions are some of those branchial motor. They do their own goofy thing again, and we'll point out just a few of those. If they have a somatic sensory function, they're going to be lateral, and visceral function are going to be in between. So what's happening is, spinal cord is pretty simple, but when we get up into the brainstem, there's a lot of other nuclei, a lot of other fiber tracts that we're not going to talk about today. We're going to talk about those over the next 2 lectures. We're going to focus today just on these cranial nerve nuclei. Additional integrative nuclei get added on as we get to the brainstem, and they tend to be added more ventrally. Cranial nerve nuclei are dorsal, unless I tell you otherwise. Okay, so I want you to become really familiar with this little diagram. You're going to see it on a lot of the slides today, because this is really going to help you. But this is basically the same thing. So when we're talking about somatic motor, and again, I told you we were going to come back to cranial nerves, there's only 4 cranial nerves that have somatic motor function. You can go back to the cranial nerve lecture, go to those tables at the very end, where it organized the cranial nerves by their function. And those cranial nerves are 3, 4, 6, and 12. So they're the three cranial nerves that control your eye muscles, and the 1 cranial nerve that controls the muscles of your tongue. Those are your 4 somatic motor. And we're going to think about where they are relative to the medulla, pons, and midbrain. Now the branchiomerics, like I said, they never play, they don't play by the rules, so they are medial, but they tend to drop down just a little more ventral. And we're not going to talk about, so it's 5, 7, 9, 10, and 11. We're not going to talk about all of those. I probably will point out 2 of those branchiomeric nuclei for you to know. If they're autonomic, they're still going to be close to midline, but not immediately adjacent. We did that, right? I'm sorry, visceral motor, yeah. The visceral motor here, the autonomic, are 3, 7, 9, and 10. Okay, let's do this again. When I say autonomic and we're talking about a cranial nerve, am I talking about parasympathetics or sympathetics? I kind of heard that mumble come down as parasympathetics, so I'll accept that. Okay? Am I talking about preganglionic parasympathetics or postganglionic? Good, that one came across real well. Okay? So, cranial nerves, 3, 7, 9, and 10. There are nuclei related to those cranial nerves. The neurons in those nuclei are preganglionic parasympathetic neurons. We're going to talk about 2 of those nuclei. Again, we're not going to go through every one of them. Visceral sensory. The only visceral sensory we're going to talk about is taste. A lot of the books will consider that as, it is a special sensory, but they'll consider it visceral because it's involved in feeding and eating. And we know that cranial nerve 7 carries taste from the anterior part of the tongue, 9 from the posterior, and 10 from the epiglottis. And then the only other visceral sensory we'll at least acknowledge are the sensations that are coming up, say, from your gut, distension, pain, or from your thoracic organs. So what cranial nerve would those be most likely related to? Good, the vagus. See, you didn't forget everything. That's great. And then finally, way out here laterally, we're going to have either our special sensory nuclei, like cranial nerve 8 for auditory, vestibular and auditory information, or some of the somatic sensory, and we're really just going to focus on cranial nerve 5. Because 7, remember, 7 just does the back of the ear, 9 does do the pharynx, and 10 does the larynx, but we won't get too concerned. I'll show you one example of where we might see those cells. Okay? So, try to get this general distribution of the nuclei down first. And then we'll worry about naming which one is which. You don't have to, you can rote memorize, but I'm trying to help you guys put it into some context. Okay, so here's another way of looking at it. The other point I'm going to make, it's not 100% true, but for our purposes, other than one nuclei that we'll talk about, for our purposes, the cranial nerve nuclei are going to be found from the open medulla, from the rostral medulla on up. We're not going to look for them in that closed medulla, where it's nice and brown, okay? One exception, and I'll show you which one that is. Okay? There's always exceptions. So again, we're going to look in this area that's sometimes referred to as the tegmentum, this green area, and they're going to be sitting in the medulla. They sit right on the kind of just along the fourth ventricle. And then this red area, these are nuclei that get added on. And they're mostly involved in some way with motor function. So we're not going to talk about them today. Rather, there's a couple of ways to approach a cross-section. You can put up a cross-section and name everything on the section. I choose not to do that. I want to focus on one set of structures, and then we'll come back to the same sections and we'll add on and review what we've already covered. I think this has proven to be a little more effective. And here we can see in this sagittal view, this tegmental area basically extends all the way from the medulla, through the pons, up to the midbrain. And what happens when we get to the basilar pons, it's like somebody took this big ball of cells and just kind of smushed it up against the tegmentum. But again, we're going to come back to that probably next time. Okay, a cross-section. We talked last time about trying to use, where possible, landmarks that we know are present on the outside of the brain that we studied in the last block. So here's a dorsal view of the medulla, because we're going to be focusing on dorsal structures today. Cerebellum has been taken off. Some of the cerebral cortex is gone. So we're just looking right down on the brainstem itself. And here's our fourth ventricle. There's the obex that kind of delineates caudal from rostral medulla. And we're going to take a cut. I'm going to take a cut right through this part of the medulla, and then I'm going to kind of stand it up so I can look at it on the screen here. Okay? Any of you working in a research lab where you cut tissue and put it on slides? Nobody? That's what we're talking about. Cutting something, and if you put it on a slide, you'd have to flip it over, right? Okay, so let's make our cut where this dashed line is. Here's my other rule when doing cross sections. The first question you ask yourself, same question we asked last block, where am I? Am I in the medulla, the pons, the midbrain, the diencephalon? And ultimately, you should be able to just look at the shape of this section and be able to say, I am in the medulla, and I know because it's open here at the top, I am in the rostral or the open medulla. If it's fairly round like this, that's the only place you can be. Okay, so let's focus up here. And we can see, especially when we're kind of close to midline, a lot of clear areas. And if an area looks clear like that on a fiber-stained section, what does that mean? It's a nucleus. And what would we find in the nucleus? cell bodies, right? Okay, so let's focus on this area, and let's try to identify some structures. Okay, here's another, I'll give you my cue here too, I forgot. Okay, you can look at the shape, that should tell you initially where you are, and then find something you can identify in the section. Not necessarily the structure that's being pointed to with an arrow. Find something. And for me, when I look here, you've obviously got this huge, convoluted looking nuclear structure, even though it's not related to the cranial nerves. You know, that's our inferior olive that formed that out pocket, that olivary tubercle, which we can see here. That's another cue that's telling you I'm in the medulla. Okay. So, here's an arrow. And it's pointing at what looks like a defined, rounded structure, and it's right on the midline. So functionally, what should you think those neurons are involved in?
Somatic motor.
Good. Somatic motor function. Now, automatically, we only have to talk about 4 possibilities, right? As I said before on that other slide, there's only 4 cranial nerve nuclei that have a somatic motor component. 3, 4, 6, and 12. Well, if you think about it, does cranial nerve 3 come out anywhere near the medulla? No. Okay, I could probably check that baby off. How about the trochlear? That's pretty rostral also, right? Because it comes off the back of the pons, wraps around. Okay, cranial nerve 6 is the abducens. That pretty much comes off a little higher, doesn't it? Between the pons and the medulla. So what are we left with? So if I eliminate 3, 4, and 6, what am I left with? Okay. I really want you guys to exercise your vagus nerve. Make those laryngeal muscles work. And 12 to get your tongue working. Okay? So, by default, and without having to necessarily memorize it, you could use that strategy to say, indeed, that is the hypoglossal nucleus. That is where the motor neurons that control your tongue muscles are located. And their axons, they're not as good on this section, but their axons will leave the nucleus and come down. There's probably a few of them right there. Remember, they come out in that pre-olivary sulcus. You see maybe just a few little pieces of them out there. I'm not going to ask you to identify the hypoglossal nerve on this particular, yet this section. Okay. And again, all of these nuclei are bilateral. I'm not going to put arrows for every one of them bilaterally. Let's move over a little bit. And I can see here a huge area on both sides. It's not right on midline. It's a little bit more lateral. So what functional component is that going to be?
Somatic motor. Somatic motor. Sorry, visceral motor.
Visceral motor, right? So again, here's our little diagram. Use that as your little cheat sheet for now. Notice my arrows are color-coded to match the diagram, if that helps. So, somatic motor on midline, and now I'm moving out to doing my visceral motor. Okay. 3, 7, 9, and 10. Visceral motor. Preganglionic parasympathetics. Given that we're, again, way back in the, we're back in the medulla, it's not going to be 3. 3 is going to be further rostral. 7 and 9, we're never going to see those nuclei. But what's a huge, which nerve would need a whole lot of neurons? 10, the vagus. Because the vagus is going down and it's providing that parasympathetic innervation, the organs in the thorax, organs in the abdomen. There's a lot of neurons doing that. So again, think logically sometimes as you approach this. And indeed, that is called the dorsal motor vagal nucleus. You can call it dorsal vagal motor nucleus, it doesn't matter. And here's the key thing, which I think will come up here in a second. The dorsal motor vagal nucleus contains preganglionic parasympathetic neurons. Those are your visceral motor neurons. But we know that cranial nerve 10 also innervates muscles in our larynx, right? And we call those branchiomeric. So those neurons do not live in that nucleus. So where are they going to be? Well, they live in a nucleus appropriately called the nucleus ambiguous, because nobody can ever find it really, unless you use special other stains. We know that they're located kind of right here. Remember I said the branchiomerics, they kind of follow their own rules and drop down a little bit more ventral. But this area right here above the inferior olive is where the neurons for cranial nerve 9 and 10 that go out to innervate the muscles of the larynx and the pharynx are located. I'm never going to ask you to identify ambiguous, but you should at least know that here's the important point. The preganglionic parasympathetics have their nucleus, and the branchial motor to the muscles of the larynx and the pharynx have a different location called ambiguous. Ready to try one more? So here, the other thing I brought in, this is a diagram that the book uses quite a bit. Again, we're looking here at a dorsal view. He changed the colors on me, but nothing I can do. I'm going to keep focusing on this little guy. So here's this green nucleus that's sitting right on either side of midline is the hypoglossal nucleus. And the nice thing about this view, it lets you know that these nuclei are not planar. They're not going to be in one little section. Hypoglossal pretty much begins at the obex and comes way up almost to the pons. So this nucleus is a pretty long nucleus going from caudal to rostral. 10, the dorsal vagal nucleus, sitting just lateral, also has that same extent. So there's a lot of neurons in these nuclei. Okay. We don't see the branchiomeric on here. Okay, now we're going to shift, and we're going to move laterally. So we've done somatic motor and visceral motor. Now, if we continue moving out laterally, the next nucleus we should see should have something to do with visceral sensory function. There's only one nucleus we have to talk about for that, and it's called the nucleus and the tractus solitarius. It's a lonely little tract all by itself sitting out there. And obviously I'm going to show you where it is on the cross-section after we talk about it for a few minutes. Like hypoglossal, like the vagus, it begins here at the obex, and then kind of extends out here. You know, it's not right on midline, it's not adjacent, it's just medial. We're moved out here laterally. And this little nucleus pretty much goes all the way up to about the mid-pons. So again, you're going to see it on many sections, potentially. What do I want you to put this in? The rostral part, that's where information from your taste buds terminate. So cranial nerve 7, cranial nerve 9, cranial nerve 10, carrying taste from either anterior, posterior tongue, or epiglottis. That information comes in with those nerves. You know what they look like now on the brainstem. Those axons are coming in, and they're going to enter what's called, or form what's called the solitary tract. The axons will leave that tract and synapse on neurons in the solitary nucleus. So where, let's say for cranial nerve 7, where would the first order neurons be located? I know this is reaching back a whole week. Well, inside or outside the CNS? Chance. Where are all sensory neurons located? See, you knew it. I'm trying to keep building these concepts for you in your head, right? So there's a ganglion related to cranial nerve 7 that's located basically around your inner ear. What is the name of the ganglion related to 7? I think I kind of heard geniculate. Faintly, okay? First order neurons that are sending their processes out to your tongue are located in the geniculate ganglion. Those axons come centrally through with cranial nerve, other components of cranial nerve 7, and those central axons will then form this solitary tract. I'm going to show you a picture of that on one of the slides here. The caudal part of solitarius, that's where any kind of sensory information coming back with cranial nerve 10 is going to terminate. We'll come back to this much more when we talk about the hypothalamus and using that sensory information to control what's going on peripherally. So, visceral from 10 is caudal, taste from 7, 9, and 10 is going to terminate more rostrally in solitarius. So again, the tract is a collection of axons, and they're surrounded by the solitary nucleus, and that's where they're going to terminate. And you can kind of think of it as a donut. The hole is where the axons are located, and then they peel off and will synapse at different levels in that nucleus. And I already said that it has a pretty wide rostrocaudal extent. Okay. I'm going to digress here for a minute, and then we'll go back to our, let me do something else here first. Just to show you where it is, and then we'll come back to it. So, hypoglossal, dorsal vagal, somatic motor, and then you see this little bundle of axons that are just kind of sitting out there by themselves? What would you think that is? All by its lonesome.
The tract is solitarius.
Right. That's our solitary tract. I always think of it as two little eyes looking out at me from these cross sections. And this I've probably seen better here. The tract is staying dark, and this clear area just around the edge would be where the nucleus is located. I'll show you that again in an upcoming slide. But we have to digress for 1 minute here and come back. We kind of touched on this at the end of the lecture last week. But let's talk now in a little more detail about the trigeminal nerve and its associated nuclei. Is trigeminal nerve motor or sensory? I'm getting both. Primarily, this trigeminal nerve is sensory. Okay? So here's our ganglion. We have the 3 branches, V1, V2, V3. The peripheral process is picking up sensations, touch, pain, temperature, pressure from their respective area of the face. And then the central processes from all of these neurons come together, enter into the closer to the brain, and form the big trigeminal nerve. So when we're looking at the trigeminal nerve on the brain, that's a coalition of all of those branches coming together. Okay, it's in a box. It means probably you should know that. Each of the divisions, V1, V2, V3, carry touch and pain information from their respective areas of the face. Now, what happens when it gets into the brainstem? Okay. Those sensations get separated. There are specific nuclei that deal with pain. There is a specific nucleus that deals with pain. There is a specific nucleus that deals with touch. And there's another nucleus that deals with proprioception. Remember, 5 also innervates our muscles of mastication. So we have proprioceptive input coming in from those. So all that information converges, and then we get it into the brain around mid-pontine levels. And what we see, all of this on this diagram, these are the 3 functionally distinct nuclei that are all getting information from the face. One of them is called the spinal nucleus. And the spinal nucleus is called that because it begins here, where cranial nerve, think of this line here as being cranial nerve 5 coming in. But the spinal nucleus begins at that point, and then it descends all the way down to the spinal cord. I'm going to show you how they get there. Right at the level where those axons enter, we have what's called the main, or the principal nucleus of 5. And that is where touch and pressure are located. Their axons, so any of the ganglion cells that are detecting touch from the face will come in, and they will synapse in the main nucleus of 5. And then there's this little skinny nucleus. Again, they all begin here, kind of at mid-pontine levels, and it ascends kind of up toward the midbrain, or the mesencephalon. And that is where proprioceptive information from your muscles of mastication or from your teeth. Believe it or not, you have proprioceptors related to your teeth. Like I said before, am I eating an almond or am I eating a marshmallow? I need to know what kind of pressure I'm putting on my teeth. And that's a proprioceptive sensation. Okay, so I'm just going to say this. This is the only sensation where the primary cell bodies are not located out in the ganglion. The mesencephalic nucleus of 5 actually contains the first order neurons. That's just something, again, I won't necessarily ask that on an exam, but just you'll read it and go, wait a minute, I saw somewhere where it said that. Why didn't they migrate out? I can't tell you that. But it is what I want you to know is that MES 5 is carrying proprioception. I'm going to come back to that one more time. Okay, so let's look at our sensory nuclei using this diagram. And let's go way out lateral here. So, right at the junction of the pons and the medulla, we're going to see what's called the cochlear nucleus. And then this other brown structure here, kind of crosshatch, is the vestibular nuclei. Again, huge, long nucleus, going way up into the pons. This red is our solitary tract, and this kind of olive green is our spinal 5. But in general, what we're seeing is that special sensory and somatic sensory nuclei are all clustered way out here laterally. We're talking about these little green cells on this diagram. Okay, want to go back to the section? Sure. You're all here. So, here's 12 and 10. So, somatic motor, visceral motor. And I already told you what that was. Anybody remember from 5 minutes ago?
Solitary tracts.
Solitary tract. And nucleus, which is kind of out there. But okay? So now we've crossed over into the visceral sensory. Let's move over a little bit more lateral. And what we're going to look now, we're not quite at the junction of the pons and the medulla, so we're not seeing cochlear nucleus yet. So we identified the solitary tract, and now what we're coming into is this one that I showed you on the other slide, this cross-hatched kind of brownish nucleus, vestibular nuclei. And the other one that we should see there, even a little bit more lateral, or at least in the same general area, depending on where we are, would be the spinal nucleus of 5. That's this cluster of cells. Very lateral. There's the nucleus, and this is called the spinal tract of 5. So, the spinal tract of 5, where would the cell bodies of origin, this is something else we need to start thinking about when we start talking about these axons. Where did they come from? Any thoughts? What if I told you these axons are carrying primary or first order sensory information from the face? Where would the cell bodies be? Pardon?
Is it goes through the ganglion?
Not through it, they're in it. Okay? These are the axons of the neurons that are in the trigenital ganglion. And in particular, These axons are carrying pain information from the face. So within the ganglion, there's a population of cells that are interested in pain. They come in and they descend. And along the way, for our purposes, spinal 5 is a little more complicated, but for our purposes, those axons then leave and synapse on neurons in the nucleus of 5, the spinal nucleus of 5. So spinal nucleus is the second order neurons. And at the end of the block, we'll talk about where they go. Questions? Okay. So visceral sensory, and then general and special sensory are going to be way out lateral. Again, you see this organization that I'm keep pushing on you? I think you'll come to appreciate it. Okay, one quick image here. Here we are at the junction of the pons and the medulla. And there's lots of stuff here, and we're not gonna talk about it right now. But notice this looks like a little appendage, little dog ear hanging over the side of the medulla. That's a hint. That is your cochlear nucleus. It looks like this little dog ear hanging over the side, and that's where your neurons that are out in the cochlear ganglion, out in your ear, enter and synapse. So those are going to be your second order neurons for processing auditory information. And then again, don't worry about this big bundle of axons. We'll talk about those later. So remember, its cranial nerve 8 has both vestibular and auditory components. The cochlear components will split off, go there, and the vestibular axons, probably this little band right there, will just come in and terminate in our vestibular nuclei. So the sensory nuclei that we're talking about, they are receiving the primary input. The primary neurons are out in the ganglion somewhere. They're sending their axons in, and they are now going into these nuclei. Further processing to come later. And again, this is probably a little better view of the spinal nucleus. And the spinal tract is always sitting out here on its lateral side throughout its course. Okay. Ready to move on? I'm not going to take as much time now as we get up into the pons. But I'm just trying to give you a way to, again, put this together, think about it, or just rote memorize. Okay, so let's move up into the pons. So again, here's this big base, this big ball of cells, and that's represented by this red area. And remember when we looked at the basilar pons, it looked like there were a lot of bands going from side to side in the gross brain. And if you look here in this section, you kind of get that same idea that we have a lot of bands just moving from side to side. And we're just looking at the ones that are closest to the surface when we have the gross brain here. Let's go up here into the green area, which we call the tegmentum. So we're talking about this back area. And again, we're going to focus dorsally to look only today at cranial nerve nuclei in the pons. We'll come back 2 more times to identify other structures. Okay, so we're basically going to talk about cranial nerve 5, or 6, 7, 5, not much 7, 5, and maybe a little bit of 4. So here's our gross image, keep that in mind. Got our big middle cerebellar peduncle here, and cranial nerve 5, just rostral to that. Okay, so basilar pons, tegmentum, and bands. So, let's make a cut just a smidge rostral to the junction of the pons and the medulla. So we're going to be in the caudal pons. And that's what it looks like. Now, does that look like the medulla anymore? I hope not. Okay? So, lots of little bands going across, which we see on the outside. Where those bands end, here's your basilar pons, here's your tegmental area, and we want to focus again on this area that's located right on the floor, basically, of the 4th ventricle, or right under the floor of the 4th ventricle. So let's come up here dorsally. Okay, don't worry about those for now. Okay, we're coming back to that. So once again, okay, there's 2 clear areas that look really close to the midline. Functional component. If I have what appears to be a nucleus and it's right against the midline, what should I be thinking? Good. I just have to phrase the question the right way. Okay? Well, it's probably not 12 because we left 12 down behind in the medulla. Do you think it's 3 or 4? No, we haven't quite got there. They're going to be more rostral, which leaves okay. Real quiet. You guys are so quiet. Use your outdoor voice. Cranial nerve 6. So, just like we did before, midline, there's your abducens nucleus. That is where the cell bodies that go out to innervate your lateral rectus muscle are located. And in fact, there are the axons leaving, and ultimately they will emerge right here at the junction of the pons and the medulla. So this is nice. On both sides, you can see cranial nerve 6 coming down here. Okay. The good news is there are some parasympathetic nuclei here, which they're very small, they're very hard to see, so they're related to cranial nerve 7 and 9. We're not going to identify them. But facial nucleus, facial nerve, motor or sensory? Motor. So, but it happens to be branchial motor, right? It's not somatic motor. So it again, just like ambiguous, it kind of sits down here, just a little bit more ventral. Over in that little corner there. Now the facial nerve, really kind of interesting. We know that it emerges here from the lateral side, just at the junction of the pons and the medulla. So here's the facial nucleus. So it would make sense that the facial nerve should just kind of emerge, right? Just pop right out where it's going there. That's not necessarily, well, it's not what happens. And I think this has to do with embryology. I think these neurons migrated and it's kind of left their axon behind. But the axons coming out of the facial nucleus actually come dorsal, and they literally swing around the abducens nucleus. It's called a janu, or bend, and then come right back toward where they began, and then go out. I don't know why they didn't just hop over and go out, but they decided to go. And again, embryologically, I suspect they were all closer together, and then the facial nucleus migrated, and its axon kind of spun out behind it. Okay? But here's your facial nerve. This is your good landmark for telling you you are in the caudal pons. When you see that nerve coursing around like that. And just to be complete, that's what's left of solitarius, because we're way up here, almost where it ends, so it's there, don't worry about it. So if that's, and now we're coming over here lateral. I see a nucleus, and I see a bundle of axons sitting just lateral to it. Did we see something like that in the medulla? And what did we call that? What nucleus might, so let's do it this way. Lateral, motor or sensory? That was kind of all over the place. Remember, motor is medial. That means sensory is lateral. You'll have to make your own little mnemonic for that. Okay? So, and I'm not going to name it one of the motor nuclei. And we talked about a sensory nucleus that began in the pons, came down through the pons, and then we were just looking at it in the medulla. And it goes all the way to the spinal cord. Spinal 5. And that's what this green little band is here. That helps. Okay. Let's come back and look at what, because this is an important concept. So we're going to do the same thing we just talked about before. Ganglion cells carrying information from different parts of the face, but they are all carrying touch, pressure, and pain from those areas. Different ganglion cells, but they come together, form a single nerve that goes into the pons about mid-pontine levels. All three come together to enter. Now there are motor axons that are in there also that are going out to your muscles of mastication. I'll show you where they come from in a minute. Okay. So, 3 nuclei. Obviously I've said this maybe 3 times, so you think it might be something kind of important? Yeah. So, sensory input is processed by 3 different nuclei. The main sensory nucleus is located right where cranial nerve 5 comes in. Touch, pressure from all 3 divisions of the trigeminal nerve. That spinal nucleus begins in mid-pons, and we're going to see it in all sections until we get to the rostral pons, sitting out here laterally. Nucleus, and then the fiber tracts sitting around it laterally, carrying pain from all 3 divisions. And there's our mesencephalic nucleus, carrying proprioception from the muscles of mastication and our teeth. And then the motor nucleus, because motor is medial, sits just medial to the main sensory nucleus. So now we have 3M. That's a manufacturer. You guys know what 3M manufacturing company is? Make Scotch tape, things like that. Okay? So when we get to trigeminal, motor nucleus is medial to the main. Okay, let's look at this one more time. So there's the trigeminal ganglion. The axons of those neurons that are interested or carrying pain enter at mid-pontine levels, and then those axons actually descend, and along their way, they're going to give off the axons will come out and synapse on neurons in the spinal nucleus of 5. The green cells are carrying general sensation from V1, V2, V3. Enter, same point, but they synapse on neurons in the main sensory nucleus. And then in the mesencephalic nucleus, cell bodies are actually in MES5, and they're sending their processes out to our proprioceptors. And then the motor nucleus, remember those are output. Those cells are leaving, cell bodies are in the CNS, and they're going out to the muscles of mastication. So, can we find those on a real cross-section? So now we've moved up. Now we're about mid-pons. I don't really see a good branch of 7 coming. I do see some nerve fibers coming in here, and we'll talk about what those are in a minute. But these nerve fibers, and what I do see are some structures, what appear to be nuclei way out here laterally. Not so much medially anymore. I see a big bundle of axons there, yet to be discussed. We're at the level of the middle cerebellar peduncle. We're coming very close to the rostral end, and these axons that we're seeing kind of looking like they're coming through it and right into the pons here, those are the incoming fibers of cranial nerve 5. Here's a nucleus that's medial to those incoming fibers. That's motor 5. The nucleus that's lateral to those fibers is the main sensory nucleus of 5. Just like I showed you on that other diagram, you have the motor nucleus, the main nucleus, and the axons coming in. MES5, don't even worry about it, it's a little dot. Would I see spinal nucleus of 5 here? No. Because if you go back to that other diagram, if you're seeing the main sensory nucleus, you're beyond spinal 5. Spinal 5 develops just caudal to the main nucleus, so you're not going to see them together. I know this is fuzzy because I had to blow it up and didn't come out as clear as I wanted it, but incoming fibers of 5, and this is your landmark. Those axons separate the main sensory nucleus from the motor nucleus. That's your key relationship there. And again, MES5 is that little dot up there. Okay. Let's take a little break who's had what's called trigeminal neuralgia, which is an excruciating facial pain. Just a breeze blowing across your cheeks sets off incredible pain. It's been called suicidal, in fact, that it's so bad. And that's called trigeminal neuralgia. And sometimes, it has to do with the distribution to that spinal trigeminal nucleus. For a long time, people didn't know what caused it, but more recently, the studies have shown that in at least 70% of the cases, or more, there are actually found to be compression of the trigeminal nerve by a blood vessel. And the pounding of the blood vessel is irritating these incoming axons. And guess which? The artery that's been found to be be the biggest culprit is your superior cerebellar artery. Now you're thinking, wait, that's going to the cerebellum, how does that affect cranial nerve 5? Let's go back to this diagram. Here's our superior cerebellar artery coming off of the basilar, and as it wraps around to come to the cerebellum, notice it passes right next to the trigeminal nerve. Pain fibers are small diameter, lightly myelinated, so they're more likely to be more irritated by this pulsation than the touch fibers, which are larger diameter and heavily myelinated. They also tend to be located more on the periphery of the nerve. So now, what do we do? How do we fix this? Well, one of the newest way to do this, you have to go in, it's brain surgery, and they will identify the artery, and they literally put a little pad between the artery and the nerve to keep it from directly contacting it. Before this was known, what they would do is they'd bring a probe, and they can bring it in through the front of your head, and there's a little hole where these branches are coming out, and they can go through one of those openings and take it into the ganglion and just ablate it. You go in and kill all the cells in the ganglion. You think, you'd get rid of the pain, but what else would you lose? And you'd lose touch sensation. But if you're getting this excruciating pain, maybe you could live with that. And then the other surgery, which I don't have a picture of, is they would come in and they'd go down to the medulla. And they would make a cut just on the lateral edge of the medulla. And what do you think they were going after? What nucleus sits right along that lateral edge of your medulla? Not the main, because we're down in the medulla. Main century is going to be up in the pons. But what's the long one? Spinal 5. What's the nucleus that's carrying pain axons? Spinal 5. And they would go in and just try to cut the tract to 5. First, I don't want anybody messing around cutting my medulla. But that was a technique that was used. And one of the things they found out by doing that it did abolish pain but not touch, which further confirmed that tract is carrying pain axons. Have a very steady hand that day, I would hope, for my neurosurgeon. Okay, last couple of minutes. Midbrain's pretty straightforward. You don't have to worry about the oculomotor nerve. I'll show you the trochlear, but we're not going to worry about it too much. So now we're going to come up here above the basilar pons, back to our tegmentum. Our landmarks are going to be these big stalks on either side, which we call the cerebral peduncles. And the space in between is the interpeduncular fossa. And there's cranial nerve 3 popping out. Mammillary bodies are not part of the midbrain. And then dorsally, we're going to see the cerebral aqueduct and the overlying tectum. Okay, so those are our gross landmarks. So if we're looking down on the tectum, we see it's made up of these 4 little bumps, which we call colliculi. Again, we're not going to worry about that per se, but if you're ever looking for cranial nerve 4, come right under the inferior colliculus. It's the only cranial nerve that comes off of the dorsal side. Okay, but so that's the nerve going out to innervate your superior oblique muscle. Where's the trochlear nucleus? Well, again, here's our cut now is coming just across the upper border of the pons, through the caudal midbrain. Here's the cerebral aqueduct. See if I have that labeled. Yeah. And that would be the overlying tectum. And we just call the area around the aqueduct the periaqueductal gray. Again, not something, I'm just going to show you that now. Okay, so let's look at that area. So there's the cerebral aqueduct, or dorsal, and right again, very close to midline. At least on this side you can see it, a teeny tiny little nucleus. It only innervates one muscle, it doesn't need a gazillion cells. But that would be your trochlear nucleus. And there's the axons coming out and beginning their course dorsally to come off on the dorsal side. That's not one of the nuclei that I will ask you to identify either. Okay, last level. Midbrain, right through the middle of the midbrain. Again, very characteristic shape. We have these 2 big stalks or columns sitting most ventral, and look what we have here. We have these 2 big stalks sitting ventrally, which we call the cerebral peduncle. So the cerebral peduncle is formed by thousands of axons, yet to be told where they're coming from. And then we have this area where there are some nuclei. Let's not worry about them now. But let's come back here into the tegmentum. And again, I see a nucleus that's right on the midline, just kind of below our cerebral aqueduct and its surrounding gray matter. Functionally, what kind of components would that have? Somatic motor. Well, we've done 12, we've done 6, we've done 4. What's left? 3. There's your oculomotor nucleus. Oops, just more landmarks. And those are the axons of 3. So the neurons in 3 are sending their axons out, and they're emerging right here in this interpeduncular fossa. You can see those on this, this cross section. Very difficult to distinguish, but this more lateral part, remember oculomotor does have visceral motor component. It's got some preganglionic parasympathetics that go out to the ciliary ganglion. And this is where they're located in what's called the Edinger skull nucleus. Okay, so somatic motor would be these guys right on the midline. Edinger Westfall is just a little bit lateral. Somatic motor, visceral motor. They're all coming out together in these in bundles of axons that are going out to the down here. You can see them coming out and going out to the eye. Okay, so we've gone through 3 levels: the medulla, the pons, and the midbrain. I hope as you look at these sections, they do look different to you, and that you will be able to distinguish the medulla from the pons from the midbrain. So that's the first question you ask. The next question, am I on midline? Am I medial? Am I kind of halfway over? Am I out lateral? And then you can begin to think about what to identify. So next time, we'll start identifying some other structures other than the cranial nerve in the eye. Again, if you took your exam on paper, I did bring the paper copies with me. If you want to look at them,